Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today is September 4th, 2025, and we're back from our summer break and starting a new season of conversations. And today we're talking to Michael Roberts. Michael is Associate Professor in the Department of Otolaryngology, Head and Neck Surgery, and in the Department of Molecular and Integrative Physiology at University of Michigan. Michael's lab studies cell types and circuits in the auditory system, primarily in the inferior colliculus, with the goal of discovering how it contributes to our sense of hearing and how the various cells that are in there interact, and the network works to produce the function of the nucleus. And he uses electrophysiological methods in situ and intact animals, and in slices, genetic cell identification and stimulation techniques, and some cool visualization methods for seeing cells and their connectivity. And we learned today that he also does some transcriptomics. So welcome, Michael.
Thank you, Charlie. It's wonderful to be here with you.
Also with us is Marina Silveira, our own inferior colliculus expert, podcast regular, and also a previous postdoc of Michael's.
Yes, that's right. I'm happy to be here too.
I'm your host, Charlie Wilson. And this is our first auditory system discussion. We talked to Marina by herself almost exactly a year ago, September 2024. And you can go back and find that one in our catalog. And we talked to Nace Golding last March. He talked about some lower parts of the auditory system. And I recommend those. Those were good. And I don't want to, revisit everything that we said before, but just for context, Michael, and Marina, since you both could do it, could you give us some big picture on auditory system and where the inferior colliculus fits in?
So I really like the arrangement of the auditory system in the brain is really interesting and quite different from other sensory systems. So basically all the information about sounds enters the brain through the two auditory nerves, and the auditory nerves synapse on these two little nuclei called the cochlear nuclei that sit on each side of the brainstem. And we always like to think when you look at them, they almost look like Mickey Mouse ears on the side of the brainstem. So all this sound input comes there and then gets sent to a whole host of different nuclei spread throughout the auditory pathway in the brainstem. And we think of this as like a big divergence in the auditory pathway. Sound information is getting sent to different processing centers, many of which have very specific processing tests they're performing. Then almost all the output of these auditory brainstem nuclei converges and comes back together in the midbrain at the brain region in my lab studies. It's called the inferior colliculus. So we often call the inferior colliculus the hub of the auditory pathway because it's a place where a lot of inputs come together. The inferior colliculus, in turn, projects up to the auditory thalamus, which provides the main auditory input to the auditory cortex and then the higher centers of the auditory pathway. So studying the inferior colliculus, we're kind of in this privileged spot right in the middle of the auditory pathway. It's the first place that all this information about sounds is being brought back together.
So presumably these early parts of the pathway are analyzing the sound, pulling out particular things like localization or pitch or volume or whatever the features or changes in frequency.
Exactly. I think the most famous example, there are a couple of nuclei called the medial superior olive and lateral superior olive that extract information about sound location, where sounds are coming from in space, and they're very specialized for that. And then there's other centers that do auditory tasks that again are specialized not quite as clearly as that, but yes, detecting things like sound intensity. Pitch is a much more complicated concept we can talk about if you want, but pitch detection, that's very controversial where that happens in the brain. Probably not in the brainstem.
Uh-huh.
Yeah.
Cortex then?
Probably.
Wow, okay.
It's a very controversial area.
Okay, so that's great. That's a great overview. So in your lab, you're basically trying to figure out how that particular set of neurons works. And I guess the obvious thing you need is a kind of schematic diagram. If I was going to reverse engineer a brain region, I would want to know what are the components and how are they connected and what are their individual properties and what are the inputs and what are the outputs. So that's what you're doing, is that right?
That's right. Or I like to think of it, this is a lot like when, for me and for many of us when we were kids, and we got a screwdriver and we decided to take apart Dad's radio because we wanted to see how it worked, right? So we're kind of doing that same thing in the auditory midbrain. We're trying to take off the cover and look inside and see how the different parts work together. And the first problem with that is even figuring out what are the different parts.
So what are the different parts?
So we know the inferior colliculus is really densely packed with a lot of neurons. In fact, there are 5 times as many neurons in the inferior colliculus as in the lower auditory brainstem nuclei combined. So in the auditory pathway, there's actually this big expansion in neuron numbers when you get to the auditory midbrain. Then, of course, there are other cell types like different types of glia supporting things in the inferior colliculus, and we know very, very little about what they're doing. But my lab's mostly focused on trying to understand what are the different neuron types in the inferior colliculus and how do they work. And globally, we already knew before my lab started that about 80%, 75-80% of the neurons in the inferior colliculus are glutamatergic neurons or excitatory neurons, and the remaining neurons are GABAergic or inhibitory neurons. We also knew some things from past work, especially from Doug Oliver's lab, about in the central nucleus of the IC, the part of the IC that gets most of the ascending input from the auditory brainstem, neurons tend to have 2 main shapes. They're either these really flat cells that were called disc-shaped cells that keep their dendrites kind of in a thin layer in the IC, and then there's stellate cells that send their dendrites across layers in the IC in different directions. And so this was kind of the state of knowledge. There's more nuance to it, but largely it's been very hard because when people look within these classifications based on excitatory or inhibitory neurons or disc-shaped or stellate-shaped neurons, there's a whole bunch of diversity and neuronal properties within those groups. So it was pretty clear we need something else to tease apart these neurons into, different functional groups.
So what is that? I mean, I might, because of the way you said it, I might say it's function that you would use to tell them apart.
So function sounds great. And I think, a lot of people tried, especially with in vivo electrophysiology recordings, they would put an electrode in the inferior colliculus, record from a neuron and play different sounds and see how the neurons responded. And so there have been some classification schemes that have come out of that. And the general problem has been anytime somebody comes up with a scheme, somebody then goes and looks more closely and finds exceptions to the rules and neurons that fall between the categories that people previously found. So our perspective in my lab now is that we think that the neuron types are actually going to be defined by their functional roles within circuits rather than their function in terms of how they respond to a sound.
Cool. That's a cool thing to say. What does it mean? I mean, what is the functional role in a circuit?
So if we think of a circuit that has excitatory and inhibitory neurons, it's maybe easier to think about from the inhibitory neuron side. There's going to be some inhibitory neurons that get input and then inhibit the excitatory neurons, provide something we would call feed-forward inhibition, right? And so those neurons that provide feed-forward inhibition, which we've seen evidence of in some of our studies, that's a specific circuit function they can provide. And so we've seen evidence that, for example, you stimulate some of the ascending excitatory inputs to the inferior colliculus, those excitatory pathways coming up into the IC will directly synapse onto some cells and provide excitatory input. But then that excitatory input gets truncated in time or cut off by an inhibitory input coming from another IC neuron that was activated by that same ascending pathway. And so you start to think about things in terms of that, and you start to see, these are like circuit widgets that we can put in different computations, like in terms of sound processing, but if you look at the local circuit level, they're working in a similar way.
So that's a little bit like the way electronic circuit designer would pick a transistor and stick it somewhere.
Exactly.
But the electronic circuit designer is thinking in the forward direction. So he knows what he wants to get on output and is putting these widgets in a way that may to untrained persons seem mysterious. But he knows what the output's going to be. So are you worried that after you've figured out the function of the neurons in this circuit way, in this way that only an electrophysiologist could love, that you now have another step to figure out how that gets you to audition?
I think there will be another step, but I think once we know these fundamentals about the circuits and how they're arranged, then we have the ability to actually probe how they contribute to sound processing in a more direct way. Like we can go in and remove a circuit element by inhibiting it, or change its, functioning by exciting or inhibiting it. And I think that will help us tease things apart. But I think the question you raise is something that keeps us all up at night and is a big problem in any sensory field, but certainly in the auditory field, is we tend, when we go, especially with in vivo recordings, we impose on the system what we think it should be doing by the sounds we present to the animal. And most of the time we present these sounds in a very artificial situation where the animal's not behaving, it's just sitting there still, and it doesn't even care about the sounds. We're just playing them and seeing how the brain responds. And the other school of thought from the neuroethology side is this is totally the wrong way to be thinking about this. We should actually be looking at how these circuits operate under more natural conditions, which of course experimentally becomes very challenging. But I think if we know what the players are at the neuronal level and the circuit level and know how to manipulate them, which thanks to all the cool toys we have in neuroscience now, we're starting to be able to do pretty well, then we can go put animals in different environments and start to test these things as we build things up. And I fully expect we're going to be surprised when we get to that point what we find.
So when you talk about the circuits, right, you think as like one neuron, they don't necessarily are only part of this one circuit, right? So this neuron in the inferior colliculus that is receiving these excitatory SN inputs is inhibiting a local neuron, which potentially is like a few micrometers away, but it might also be projected to thalamus at the same time, or to the contralateral IC, or even like within subdivisions of the IC. So how do you think about a same neuron comprising multiple circuits?
I think that's maybe going to be the biggest challenge, is that we are starting to find pretty clear evidence that single neurons in the IC can have axon projections that are very local, make synapses onto neurons in the neighborhood right by the cell, but also then go farther than the IC and synapse on other neurons, and then may project to the other side of the IC on the other half of the brain altogether. And that same neuron might have an axon that branches and goes up to auditory thalamus. And so, thinking in terms of how all of that wiring diagram ends up influencing function is I don't know how to think about it yet.
There's no modularity. I think it's frightening, because when we're imagining things that are like imagining something that's fixable, you would say, well, this piece has got this job and this piece has got that job, and when something goes wrong with that function, it's that piece, and you can sort things out by pieces because it was designed to be modular. But it seems like the auditory system is designed to not be modular. Everything kind of connects to everything else. Things that are up the pathway quite a ways project all the way down to the very beginning of it. So in some ways, this is not unique to the auditory system, of course, but I worry that you can't understand any of it until you understand all of it.
I suppose that could be the case, but I think the complexity of the auditory system, at the same time, it may not be modular the way we're looking at it right now. But I kind of suspect when we get to a point where we understand the circuits better, there are patterns there. It's just probably harder for us to see, especially in a lot of these auditory brain regions in the midbrain brainstem. They're evolutionarily very old. So they've had, they existed in much simpler animals that had a lot of evolutionary pressure to keep the brain small and compact and energy efficient. So I suspect they're like there's a circuit architecture that's just, we're not seeing it because everything is jammed together and mixed together in a complicated way. But it's almost like if we could expand it out and see what the network map actually looks like, that map might not be as bad as it seems.
So can we find some encouraging examples other places in the nervous system where people have completed this task, end up with their diagram and said, oh, look how great it was to have that. Look how powerful it is to know that stuff.
So I don't know if there's anywhere where it's been completed, but certainly in the neocortex, we know a tremendous amount now about neuron types. In fact, it seems like every year somebody comes out with a new study where they can subdivide things even further. So we know there are many different types of pyramidal neurons within a single layer of neocortex. There can be multiple types of pyramidal neurons. We know there are a whole host of different types of inhibitory interneurons. And what's cool is in there, because this has been studied so well, we know the connection tendencies among these quite well. And people have been able to go in and manipulate single neuron types and see how that affects things like animal behavior in a task. And it's really remarkable. You can change activity of one neuron type in the cortex and have behavioral effects. And so people now are even looking at, can we modulate activity of this neuron as a treatment, potentially, right? And so I think the power of that has really been shown well in neocortex. And I think if we can bring that to other brain regions, hopefully we can have interventions.
So when they do that stuff in the cortex, or when you're going to be able to do it, I mean, for a long time people have just stimulated some part of the brain and they see something happen and they go, that's what happens when you stimulate that part of the brain. But I would ask them, if I had a chance, how does stimulating that cause that?
Yeah.
Right? I'd like an intuitive understanding of that. Do you think they have that kind of understanding about the cortex?
I think in these highly interconnected networks that we have in the brain, I think these are very challenging issues. So causality is a big question, right? We know we can go change activity in one neuron type and see something changes. How it caused that change, I think, is a much harder question to address. And so in the inferior colliculus, for example, I'm hoping in the next 5-6 years we're at a point we can manipulate specific neuron types and see that they make something change. But I also suspect how they make something change is a very complicated question. I mean, there's beautiful stories in cortex, for example, of paradoxical effects, like you inhibit inhibitory neurons, you actually get an increase in inhibition, net increase. This idea of an inhibition stabilized network that there's been a lot of beautiful work on. And so I think the big risk in neuroscience is, I think as scientists, we like to follow our intuition, but I suspect our intuition isn't up to the task a lot of the times now when we're dealing with really complex neural networks.
So sometimes, even if our knowledge is imperfect, we can discover something that's useful in the world. And so it seems like the auditory system is a place where people have been able to discover useful things, and we have things, implants that give people back their hearing and that sort of stuff.
Absolutely.
And do you look for applications? I mean, what is the inferior colliculus for? What happens if it goes wrong? And could we fix it?
So what the inferior colliculus for is a really deep question, and I don't think we have a good answer for that other than we know if you lesion the inferior colliculus, most behavioral responses to sound are gone. Like in the animal or human can still tell there's a sound that exists, but they can't identify the sound, they can't tell where it's coming from. There's certainly no speech perception, things like that. So we know the inferior colliculus is very important. We get very lucky in science because sometimes, even without a full understanding, we're able to accomplish amazing things. So like in the auditory field, as you were mentioning, the cochlear implant is an amazing device that frankly, as scientists, we got insanely lucky that it even works. I mean, if you think about it, in some ways, when it started out especially, it's a very crude device. There's a handful of electrodes that get strung along the length of the cochlea, and that's enough to restore a sense of hearing in a lot of people, with a lot of processing that determines how those electrodes are activated based on sounds. But that's something that actually a simple solution ended up working remarkably well. And so there's a hope, I certainly have a hope that we may not have to fully understand something like the inferior colliculus to develop an intervention. And I think if we know just a bit more than we know, we might be at a place where you could have an implant into the inferior colliculus to help restore hearing for people who, for various reasons, may no longer have a functioning auditory nerve. So a cochlear implant's not possible for them.
Where does object identification happen in the auditory system? Is that inferior colliculus thing?
I think,
like when you say speech.
yeah, no, I think in the inferior colliculus you certainly have neurons that encode for different speech sounds. Mm-hmm. And there's some evidence in rodents that you can have neurons that are quite selective for specific vocalizations. But these are, like if you're dealing with mice, this is a very limited vocalization repertoire. So I think like real coding for speech, like identifying words and all of that's happening in the level of the cortex much higher. But of course, if you have things going wrong earlier in the auditory pathway, then you won't even get the information up to the cortex to be able to do that.
There, in the visual system, there's something called blind sight, in which the superior colliculus can drive behavior even when the cortex is gone. Is there anything like that? And
there are some studies, right, where you silence the cortex or use some technique to, the auditory cortex, or even like aspiration of auditory cortex. You still have some auditory encoding being properly done at the level of the inferior colliculus.
Yeah, I think from an evolutionary perspective, I think the inferior colliculus mediated a lot of very, fundamental survival behaviors related to sound. Like the inferior colliculus has pretty strong projections to periaqueductal gray circuits that regulate things like freezing and defensive behaviors in a situation where a mouse or an animal is going to be afraid of something. So I think inferior colliculus has connections in that way. It also provides auditory input to the deep layers of superior colliculus, where you actually have this really cool integration of visual and auditory information that drives head orienting responses. So if there's a sound coming over from the left and you turn your head to that, the superior colliculus was probably involved in making that happen. So I think without cortex, you can still have certain auditory behaviors that are higher than a reflex level, but certainly not probably the same rich repertoire you get with the cortex present.
Higher than reflex level. Wow, I don't know what that means either. But
so there's this idea in the literature of something called a fixed action pattern that's a stereotyped behavior that's more complicated than a reflex. So the famous auditory...
So turning my head, is it one
that
of
would
those?
be like I think that's a fixed action pattern, yeah. So the auditory reflex is the acoustic startle response, right? So there's a sudden sound you weren't expecting and you jump. And that's a reflex arc that doesn't have to go to the inferior colliculus. The inferior colliculus can influence it because a lot of the times we can suppress it, obviously. But it doesn't. It's a very interesting reflex arc that involves neurons that sit along the auditory nerve itself and project into brainstem to directly drive motor responses. So I think the IC is regulating things more complicated than that.
Okay, well, sounds you're being very cagey about it because I guess we don't have the behavioral experimental
We don't
data.
have a lot. There's some evidence. There's a study from, I think, George Brees in 1984 showing that if they electrically stimulate the IC, a mouse or rat, I forget which, a rodent will run. And depending where they stimulate, the rodent might, if they stimulate dorsal IC, the rodent will run for a lot longer than the electric stimulation lasts. If they stimulate ventral IC, it'll only run while the stimulation is going, basically. And then more recently, Li Zhang's lab at USC showed they can use optogenetics to drive running behavior. Basically, you excite pathway from the IC to periaqueductal gray. It drives running. So I think at least there's pretty clear evidence of that. There's also some evidence there's some of the periaqueductal gray circuits are involved in driving vocalizations for simple vocalizations, and that the IC might be contributing to that. So I think there's some basic behaviors that it's regulating.
So as we take apart the circuit of the inferior colliculus, and you find however many, 52, exactly 52 excitatory interneurons, and 25 inhibitory ones, and the excitatory ones all project out, and the inhibitory ones do too, because that's the worst case scenario, and I think about worst case scenario like that. And we learn all the connectivity of them. What's next? I mean, let's say we wrote that. We could write that diagram out right now and have the synaptic organization of the inferior colliculus complete.
So what I'm really excited about long term, there's a lot of directions we can take this, like trying to understand how these circuits change in hearing loss, how they start to function to restore hearing when somebody gets a cochlear implant, right, which is a very different stimulus to the auditory system, but amazingly the auditory brain can make sense of it. But there's another side to this that I'm personally very excited about, because I think it's poorly understood area that affects a lot of us. That we lump together this set of disorders into something we call central auditory processing disorder, which is basically a catch-all for, we think there's a problem in auditory processing in the brain that's affecting somebody's ability to understand speech in an environment like a restaurant that most people are fine in. And as far as we can tell, everything's fine in their ear. So this is probably in the brain. And so I really suspect that some of this is happening in the IC, that there are differences in how the circuits wire up and function. And if we understand how the circuits normally work, we might be able to go in and say, this person who, like me, who has a really hard time understanding someone speaking to me in a restaurant, it might be because my IC has this change in its wiring different from other people, and maybe there's something we could do to help with that. Or also, when you look at cochlear implant patients, there's a huge diversity in how well they respond to cochlear implants. So if you control even for people who have very similar damage to their periphery and had similarly successful cochlear implant surgeries, some do amazingly well. And within months are understanding speech and functioning at a very high level with their implant. Others don't do well, and we don't know why. And I suspect that's probably due to plasticity in the brain and how circuits are adapting to this new input from the cochlear implant. And so I'm also very interested in, if we understand how the circuit's normally working, we can find out how we can drive changes in it to support rehabilitation when somebody gets a cochlear implant.
Very cool.
That's very cool idea. I wonder what kind of, I know you use optogenetics to stimulate neurons. Is that what you're envisioning when you're thinking about drive changes?
Yeah, I mean, there's different possibilities. One possibility is maybe we do have to use something like optogenetic tools and then some sort of implant in the inferior colliculus to directly modulate the circuit function there in a certain way. But the other thing is the IC is a very plastic place as far as we understand. There's a lot of, the circuits can change and learn. And we know the inferior colliculus gets a lot of neuromodulatory input from different modulatory systems like cholinergic input, serotonergic input, dopaminergic input, and others. And so maybe there's some evidence that the cholinergic input, for example, drives plasticity in the IC. So Nobusuga had some beautiful studies in bats in the early 2000s about this, showing this really important role for cholinergic signaling through muscarinic receptors and driving plasticity. And so maybe it's also the case that we just need to learn, we drive this neuromodulatory input to the IC while somebody's doing a rehabilitation task, and suddenly they learn it better.
So one of the treatments for tinnitus that I've read about is this coordinated reset stimulation. which I think grew out of the deep brain stimulation literature, as people are trying to find the ideal pattern for deep brain stimulation. And about this, this is like making these patterns of sounds, and you listen to them for a long time, and it's supposed to drive plasticity in your auditory system to combat tinnitus.
Just external sound.
It's just a sound, yeah. You can buy an app for your phone, and it will play these sounds, and you can tune it to the frequency of your tinnitus. So is that, what about that? I mean, because that's the easy kind of stimulus to give, is just say, you've been listening to the wrong kind of sounds. You've been listening to loud rock music, way too loud, or you've been listening to big explosions because you worked in a place where there were a lot of explosions. And so that's killed some cells and driven some plasticity, and now we could try to give you a stimulus that would move the plasticity in another direction.
So
I know this is putting you on the spot.
No, it's okay. So the tinnitus treatment field is very controversial space.
Complicated.
And complicated.
Very
I will say, as far as I know, there's no strong clinical data that just a sound treatment can treat tinnitus. The one treatment I know of that has very strong evidence supporting it was developed by Susan Shore at the University of Michigan, and it took advantage of spike timing dependent plasticity, a form of neuroplasticity that was first shown to take place in the dorsal cochlear nucleus in brain slice work from Thanos Giannopoulos and Larry Trussell. And so the dorsal cochlear nucleus is this interesting place, early station in the auditory pathway, gets input from the auditory nerve, but also gets a lot of somatosensory input, including input from the trigeminal nerve, so relating to things on the face. And so it turns out the circuit, you can drive neuroplasticity in it by combining somatosensory stimulation and auditory stimulation.
That's exactly the kind of thing.
That's what you're talking. Yeah, so if you do this, if you know the right timing relationships between those, you can drive either long-term potentiation, which we think would make the tinnitus worse and you don't want, or you could drive long-term depression, which helps reduce the response of these neurons and make them hopefully reduce the tinnitus percept. And so Susan Shore's had a device in clinical trials that actually does exactly that. It pairs sounds with a little electric shock to the face that affects the trigeminal nerve, and they've had very promising results. There was a very nice paper in Science Translational Medicine a few years ago, and now they're trying to commercialize that.
So, as I'd like to kind of reconstruct the discovery pathway that leads to that, and how much did you have to know about circuits to do that? It seemed like what you needed to know about was synaptic properties to do that.
So I think it actually took, so this is a great example of what you needed to know the circuit, because it turns out the dorsal cochlear nucleus circuit is a lot like the circuitry of the cerebellum. In fact, the dorsal cochlear nucleus is called a cerebellum-like structure. And so it's been shown that there are certain combinations of activity in the cerebellum. You stimulate parallel fibers and the climbing fiber providing input to the Purkinje cells in the right patterns, you can get long-term potentiation or depression. And so the Trussell lab, in work that Thanos Giannopoulos did, they looked at these spike timing pairing paradigms by activating the parallel fibers that bring somatosensory and other multisensory information into the DCN, and the auditory nerve fibers in different patterns. They found this very tight relationship between which input was stimulated first in time drove the type of plasticity you got. And so the idea started from very basic brain slice electrophysiology work that required some knowledge of the circuitry. And then it grew into Susan Shore's lab, took this in vivo and started doing all sorts of very cool experiments to look at how they could drive this plasticity in vivo, and found this potential treatment approach.
Great.
Yeah, and
I love that.
I also think understanding the circuits, right? They may respond different to certain neuromodulators, such as acetylcholine, for example. So if you think in a line of treatment, and if you can target different circuits, it may reduce the side effects of medications, for example. if you know which circuits are contributing to what. So,
most of my life, people have told me, what you're doing, trying to work out the circuitry of the brain, isn't going to work. The circuitry of the cerebellum was understood in 1965, and a great book was written about it. It took the world by storm. Everybody read it, and we were left not knowing anything about how the cerebellum worked after that was over. And maybe that's still true, but it sounds like that book helped the auditory system.
Absolutely did, yes.
So great. I think that's a great story about why we should try to understand circuits of the brain, why it's worth it to do the hard work of finding the cell types and connectivity, even though it may not be that immediate panacea, but it isn't a waste of time to do
never.
Right.
Okay, thank you very much.
Oh, thank you, Charlie. Thank you, Marina. It's a pleasure. Yeah.
Super fun.
That was great. Thank you, Charlie. Thank you, Michael.
And this has been Neuroscientists Talk Shop.